Welcome and good afternoon, everyone. Thank you so much for joining us today as we are joined by our colleagues over at Avidity Biosciences. If you don't know me, my name is Rachel Schrader. I'm part of the care team here at Parent Project Muscular Dystrophy, and I am thrilled to be joined by our colleagues at Avidity Biosciences, who are going to share some information on the top line and functional data from the del-zota Explore 44 and Explore 44-OLE. We're thrilled to have this conversation today. I think, we've seen some of the data along the way that's just been incredibly exciting and encouraging. And there's quite the appetite across the community to learn more about the current pipeline with del-zota. So with that, I think we're just going to go ahead and hand things right over to our speakers. We are joined today by Beth Henriksen, who's the Director of Patient Advocacy, and she's going to be joined by Dr. Aravindan Veerapandiyan, who is an associate professor at Arkansas Children's Hospital, as well as Hussam Yunus, who is the DMD Global Program Head. So with that, I'm going to go ahead and hand things over to Beth to get us started.
Thank you, Rachel. Hello, everyone. As Rachel mentioned, my name is Beth Henriksen, and I am part of the advocacy team at Avidity Biosciences. We're thrilled to have you join us today as we present the del-zota Explore 44 OLE top line and functional data, which is the first of its kind. Before we begin, we'd like to thank each study participant, so the boys and men living with Duchenne, amenable to exon 44 skipping, their families, the investigators, and their team for their time, commitment, and continued contributions to our studies. Thank you. Before we get into our content, I'm going to cover a couple of upfront slides, starting with a disclaimer, and that's just to make sure that you as the audience are familiar with a few areas. So we'd like to make sure you're aware the presentation today discusses a clinical development program for an investigational product, meaning it has not been approved by the FDA. So therefore, conclusions regarding the efficacy or safety cannot be drawn from this data. The data is being shared for educational purposes and cannot be used to make any individual treatment decisions. The data presented today is from a phase 1 and 2 study, which is evaluating the safety and dosing of the product we refer to as del-zota. Phase 2 of Explore 44 is an ongoing open label extension study, which is evaluating the safety and tolerability of long-term dosing of del-zota. So once again, del-zota is an investigational product not approved by the regulatory agencies, and its safety and efficacy have not been established. You can expect us to cover the topics listed on the left-hand side of your screen. First, we'll start with a brief introduction to Avidity to include an overview of our science. Next, we'll talk about exon skipping as it relates to Duchenne, and then specifically to del-zota. We'll certainly cover our functional data that we're all gathered here today to hear about, and then we'll make sure there's plenty of time at the end for questions and answers. We know questions were submitted as part of the registration process, and we encourage you to enter questions into the chat during the course of the presentation today. The presentation will be given by the two individuals on your screen. On the left-hand side, we're joined by Dr. Panda, who's the Associate Professor of Pediatrics at the University of Arkansas Medical Sciences and Arkansas Children's Hospital, and then also by Hussam Yunus, who's the Duchenne Global Program Head at Avidity. Thank you both for being here. To get us started, I'll turn it over to Hussam for the introduction to Avidity.
Thank you, Beth, and good morning or good afternoon, everyone. So really, I'd like to start off with the vision of Avidity, which is, simply to improve people's lives by revolutionizing the delivery of RNA therapeutics. And at this point, we're really focused on neuromuscular disease, and I'd like to kind of go through with you some of the key aspects of the technology and the proprietary science that we're moving forward and studying. So first of all, in terms of the platform, really what we're utilizing here is two known molecular entities that each are well established in the field. One is monoclonal antibodies and their specificity to target receptors in this case, as well as oligonucleotides, which are very specific molecules that can target RNA to treat many different diseases. And in this case, we're really conjugating the two such that we are utilizing the potential of each to go after diseases where there has been very little opportunities up to this point. Next slide. So in the case of del-zota and the antibody oligonucleotide conjugate platform that Avidity has developed, or AOC, we're really utilizing the antibody to target muscle cells, both skeletal muscle as well as heart. In this case, the antibody binds to a specific receptor called transferrin receptor 1, TFR1. Once the antibody binds, essentially the whole molecule gets internalized within the muscle cell, at which point the oligonucleotide gets liberated and then can migrate into the nucleus, where it specifically binds to a gene that then modulates the activity. In the case of del-zota, that activity is essentially on the DMD gene, where it can promote an increase in dystrophin production. And I'll go through that in a little bit more detail in the next few slides in terms of how this happens as it relates to promoting exon skipping. So as many of you know, dystrophin is the largest gene in the human body. And it's really constructed as a puzzle, so it plays a very important role in muscle integrity and function. So you can see here that there's kind of different parts of the gene that are called exons, and they're kind of numbered here from 1 all the way to 79. And in the case of DMD, as several of you may know, and next slide, Beth, there's essentially mutations and deletions in this DMD gene that cause the puzzle essentially to be disrupted. And this is an example here where if you're just taking a snapshot of that entire transcript, and in this case, we're really kind of zooming in on these specific exons here that span from 41 to 49 in this case. And you can see that there's a missing deletion here of exon 45. In this case, the puzzle essentially doesn't fit together. And that leads to low or very little production of the dystrophin protein. So what we're trying to do here in terms of del-zota is to be able to skip exon 44. And when you skip exon 44 from the transcript, you're recreating the puzzle. And essentially that allows for an increase in dystrophin protein. It's a shorter, truncated protein. And despite that, there's still some meaningful functionality. And in some cases, there's very robust functionality, despite the fact that the dystrophin protein is still truncated. So next slide. So this is kind of, putting it all together in terms of amendability to exon 44 skipping and what does that really mean? So as you can imagine, there's a lot of different mutations that could happen on the dystrophin gene. And depending on the mutation that you have, you may be amenable to, in this case, exon 44 skipping. So there's over, I believe 46 different types of mutations. Despite that, they all may be amenable to this type of therapy if you're skipping exon 44, because you're still able to now, to some degree, fix that puzzle and that dystrophin mRNA to start making a functional dystrophin protein. So depending on the mutation that you have, you obviously may have a different length of that dystrophin that's produced with del-zota. Next slide. All right, so I think now I'll transfer it over to Dr. Panda, who's going to go over the EXPLORE44 and OLE data. Dr. Panda.
Thank you, Hussam. Thank you, Beth. Thank you, Rachel and PPMD team. And hi, everyone. let's kind of talk about the functional data from the Explore 44. So you're going to hear about the Explore 44 and then Explore 44 OLE. OLE is an open label extension. The first Explore 44 study is a randomized double-blind placebo-controlled study, right? There are actually three cohorts, two different doses, and then the placebo. And we did biopsies in all of those cohorts. And the two doses that were studied in that study was 5 milligram per kilogram every 6 weeks, and then 10 milligram per kilogram every 8 weeks. And there are a total of 19 patients who received del-zota and 7 who received placebo. Now, after 3 doses, they were eligible to roll over into what we call the open label extension. And we're going to focus on that today. The open label extension, again, based on this Explore 44 study, we decided the dose on 5 milligram per kilogram Q6 weeks as a selected dose, and all of the participants were transitioned into that dose in the open label extension. 16 additional participants were also enrolled. Next slide. So if you look at how, there were a total of 39 participants in that Explore 44 open label study. Their ages were between 7 and 27, with a mean age of 13 years. And about 70% of them, 67% of them were ambulatory, and about 33 were non-ambulatory. And majority of them, 90%, were on steroids. And again, today's update, we're going to talk about the participant treated in the open label functional data at approximately 1 year. So out of the 39, we have about 17 patients that had this 1 year mark data. And this data cut was as of June 2025, if I remember correctly. And looking at the safety and tolerability part, I think most of the side effects were mild or moderate, and there was only one participant that discontinued the study due to hypersensitivity reaction, due to allergic reaction to the medicine itself, and that was only serious adverse event that was related to the study drug, and most of them were mild. All right. I think it kind of gives you a pictorial example of how the dystrophin may look with del-zota. I know Hussam went over how the exon skipping happens. If you look at the top, that is your naturally occurring full normal dystrophin, right? When you take a kid with the exon 45 deletion, when you use the del-zota, this is how the second dystrophin is how it will look. And then microdystrophins are even shorter than what the actual dystrophin from the exon skipping would look like, which is the third one, is microdystrophin. Again, just showing that we are near full length dystrophin from del-zota. It retains the key functional domains of the gene itself, so you can have better protein. All right. Now, this is kind of before we get into the functional measures itself. So kind of see how much dystrophin was made from these in the first trial, in the placebo-controlled trial. If you look at these two groups, the 5 milligram per kilogram as well as the 10 milligram per kilogram groups, there were biopsies done after 3 doses. And if you look at the post-treatment biopsies, there is in the 5 milligram per kilogram group, there is additional 25% of increase in the dystrophin, and then the 10 milligram was 26%. So both doses, it was similar, and that's what they ended up choosing, the 5 milligram per kilogram every 6 weeks. Now, in the right side here, you can pull up the other one too, Beth. So these are the dystrophin positive fibers in one of the patients in the study. If you look at the baseline, they may have some. These red lining on the, and if you do the red circles, those are the dystrophin stain of the muscle cells. You can see how much, after the treatment, there's much more of those red lined cells, again, showing there was dystrophin expression, and it was actually present on the muscle cell membranes where we wanted the dystrophin to be. Now, along those same lines, I know we probably looked at this multiple times about the CK, creatinine kinase levels. And this is, again, an impressive data they're looking at here. The creatinine kinase levels were reduced by 80% compared to baseline, and they were sustained at the near normal levels. If you look at the graph here, you see these dotted lines right above that. So that's the normal, upper limit of normal for CK. And the blue dot ones were the ones who received del-zota in the first study. And as you could see here, there was sustained reduction of CK, and they were followed up through 16 months. We have that CK reduction kind of stayed the same. Now, the gray bars, I want to pay attention, pay attention to the gray ones. They were placebo in the first trial. When they moved on to the open label, they started receiving del-zota, and you could see the rapid reduction in the CK once they start receiving the drug, going back to the near normal levels and stayed the same. So 50% of participants had CK levels within normal range at 1 year of treatment. We'll look at now getting into the functional data. So now we know this is an open label extension study, like all of those patients were on del-zota. So we wanted to compare those with the natural history. What does it mean is his natural history of patients with deletions that would be amenable for exon 44 skipping without the medicine. How would that look? And comparing their functional outcomes with the ones who received del-zota. So we used a data set called PRO-DMD. It's a natural history study. It was observational. When we look at the whole total number of 269 patients, about 22 of them matched the criteria for exon skipping, for exon 44 skipping, and then they were also matched to the participants that we used in the trials, in the trial itself. So of course they have to be exon 44 skipping. That's between 7 and 27 years old, and they have to be on stable steroid for at least a month. And then the assessments that we looked at were some of the time tests we'll talk about. Can we bring in the other column too, Beth? So if you look at here is some of the characteristics of our natural history participants as well as the del-zota participants. If you look at the age, they were the same age almost, right? And if you look at you can look at some of the other functional testing, the 4-stair climb, 10-meter walk, and all of that. And if you pay attention to those, actually the ones in the del-zota group were much more worse to begin with compared to the ones we picked up from the natural history. It's important to keep in mind because you would expect these patients in the del-zota group to decline even faster compared to the natural history ones if you look at their baseline scores. All right, next. And then I also want to just briefly touch upon how are we going to show the results here, so to keep you all oriented to that. If you look at some of the time tests, we call time tests, the time to stand, meaning how long you would take to stand up from the floor, or to the 4-stair climb, how long you take to climb the 4 stairs. Because the shorter the time is better, right? If you do it within certain period of time, you want to do it faster. So how this is going to look when we look at the results is if you see negative changes, like if it is -2 or -4, it's actually better. Because this -2 from the baseline, -4 from the baseline, so they're doing it faster. So this is how we're going to look. You're going to see the changes in these. All right, next slide. Now, if you look at the natural history itself compared to just under the natural history standard of care, like it said before, you would expect these patients to actually decline these scores, the PUL scores or time to rise, 10-meter walk test, all of them, you would expect them to decline. And this is from the PRO-DMD natural history study. All right, next slide. All right, we're going to now look at our functional data here. It is at 1 year, and then compared to natural history at 1 year, right? So we have, for this one, it's a 4-stair climb, as you could see here. The green, the ones who are treated with del-zota, which is the blue ones, they have less time to complete. So compared to your natural history data. All right, next one would be, so this is the 10-meter walk run test. Again, you see the blue dot on the right-hand side. Again, they're improving. They took less time to complete the 10-meter walk run compared to your natural history. All right, this is the time to rise from floor. Again, improvement in the treated participants. As you could see here, the blue dot again towards the right side, then the negative side, meaning they took less time to complete the task compared to the natural history patients. Again, it's all thumbs up. Next one. Now we're going to look at the here is more skills, meaning more of North Star ambulatory assessment, right? So here, the NSAA is something that we assess, more motor skills. And as you all familiar, it's about 17 items. It has 34. 34 is your maximum score that you can get. So here, the higher the score is better, right? So if you see additional points in the scores, then it's actually better. So it's slightly different than what we looked at before from the time test. All right. So in NSAA, here more, if you look at the graph again, there was no change, which is it's a stability, indicating the stability, right? Like we talked about before, these patients to begin with were actually much more severe than the natural history participants, and even stability or no change is actually a win. It's actually meaning a good thing here too. So next slide. So this is performance of upper limb. Again, this is also a functional assessment, mainly looking at the upper limb function. Some of you might have had it done in the clinical trials, also done in the clinics. It's usually in the clinics done for the non-ambulatory patients, but I think now we are changing the practice to also include this in the ambulatory patients as well. And as you could see here, this is also the higher the score, better. And at the end of 1 year, the del-zota treated patients, it's increasing score again compared to natural history. It's a thumbs up. Good. I think in total, there was statistically significant, if you look at both the biomarker data, right? So you have dystrophin, statistically significant dystrophin increase, and there were sustained improvements in the CK, meaning your CK has been down to close to normal in patients. And then that kind of also translated into your clinically meaningful functional endpoints that we looked at, including a 4-stair climb or 10-meter walk test, time to rise, or pulse score. And then the safety profile was also, this shown to be more safe and tolerable in these patients. All right, next. I think with that, I'll hand it back over to Huzam to talk about future.
Thank you, Dr. Panda. So as you saw with the functional data, we're really, really excited about the potential for del-zota to really be potentially a foundational treatment for DMD patients who are amenable to exon 44 skipping. So the next steps around the del-zota development program is that we're working with the FDA, and we have alignment with them around pursuing an accelerated approval pathway. And in that light, we are targeting a BLA submission in 2026 to enable that potential for accelerated approval next year. And at the same time, we are also thinking through our plans around development outside of the US, as well as pursuing other exon skipping programs. The one that is most advanced for us at this point is a AOC 1045, which is a AOC that's targeting exon 45 amenable patients. And we look forward to sharing more information on that program moving forward. At this point, it's still in the preclinical stage. So we'd really love to connect with you and stay in touch. Feel free to scan this barcode, and that will be a nice conduit for you to get in touch with our patient advocacy group. So I'd like to really end by thanking really everyone in the DMD community for working with Avidity on this program specifically, as well as PPMD for hosting this webinar. It's been really a great collaboration up to this point. Again, want to reiterate Beth's earlier comment around thanking the trial participants, their families, all the investigators, and really all of the sites. It's an effort that requires all of us. And so on behalf of Avidity, I'd really like to thank the community. I think we're can open it up to the Q&A. Rachel?
Great. Thank you so much, Beth and Dr. Panda and Hussam. That data is it's really incredible. At this time, we're going to go ahead and pivot to our question and answer session.
And hi, this is Caitlin Matoso, Chief Business Officer with PPMD. I'm also on the line, and apologies, Rachel. If I may, can I just interrupt and maybe we could perhaps start our Q&A based on the exciting news that we saw this morning in Avidity's press release. I've been helping monitor some of the questions that have been submitted and come in. Team Avidity, if it's okay, would we be able to ask just a few questions regarding the exciting announcement that you made this morning in the press release?
Yes, absolutely.
Okay. Well, in that case, I will take the liberty. I think fully recognizing that the announcement is hours old and you may not be able to answer everything here today, we will try, however, to get as much information as we possibly can. So let's start broad and then bear with me if I ask some more questions that you may not be able to answer directly. So let's start, Beth and Hussam. What does it look like? We've read the term managed access program, but can you talk to us a little bit about what it means to access this managed access program and what that might look like for families who are reading and waiting with bated breath about this?
Yes. So hello, everyone. And to anyone who maybe hasn't seen the news yet, today Avidity has announced that we'll be doing a managed access program for del-zota, which opens up early access to the investigational treatment. We're really honored to be able to provide this pathway for the community and people amenable to exon 44 skipping. And we really recognize the significant unmet needs that exist and the importance of time and urgency, and that all went into informing this decision. So we've heard a lot of this from you directly and in families in the community. And so we're really just very, very excited to bring this to everyone. And perhaps, Caitlin, I can spend a second or two talking about what a managed access program is at a high level, and maybe that would be beneficial too.
Yeah, I think that would be super helpful.
Okay, so a managed access program, or sometimes it's referred to as a MAP or early access or expanded access, is a program that allows eligible people to receive investigational treatments like del-zota before approval by the FDA. And this pathway is typically for serious conditions with no or limited treatment options. So for Avidity and the MAP that we've designed, it's designed to provide access to del-zota for individuals who meet MAP criteria and whose doctor determines that their patient might benefit from this investigational treatment in the time prior to del-zota being approved by the FDA. Hopefully that's helpful a bit.
Yeah, I think that's super helpful background. And if I may, so I'm looking at the press release, and can you just, again, and recognizing you may not be able to answer everything in detail, so can any participating healthcare provider can work with an eligible family to enroll in this program? Like, what does that mean?
Yes, it's a great question. So anybody who's living permanently in the U.S. can access this program with a U.S. physician, a U.S. doctor. And the best path for accessing this program is to contact your doctor. Additional information will be available, such as eligibility criteria, on clinicaltrials.gov in the next few days. We just really wanted to make sure this news reached the community as quickly as possible with all the excitement around it. And something I would like to mention is this is led by your doctor. And this is following a very specific protocol, which means that your doctor is going to have to submit paperwork for this process, and it gets reviewed. So in that, we just like to set an expectation that it'll take some time for the paperwork to get processed. So we're announcing the program today, and it may take, let's say, up to 10 weeks if eligible to receive the first dose of del-zota.
Perfect. Okay, so up to 10 weeks after your healthcare provider sort of starts the process. And then Again, circumspect by year end, and I know more information will be available on clinicaltrials.gov imminently. Do you have any idea of when the program might start?
So it's open. It's open now, and so you can contact your doctor, and the doctor can follow the protocols and the pathway. We're working with a partner that your doctor can contact to initiate the process, start the paperwork. So it is officially open now.
Great. I appreciate that clarification. And again, I know inclusion criteria is forthcoming. Do we have any idea of sort of lower age range or upper age limit through the program, or is that yet to come?
Yes, the age range starts at 6 years old, and that was determined by the population that we've studied to date. So it's ages 6 and up.
Okay, bear with me just a few more. In terms of prior exposure to other therapies, so families who may be on either an approved or investigational product today, worthwhile for them to reach out to their clinicians if they have an interest? Are there any barriers to anybody who has prior exposure to therapies in the MAP?
Yes, please do reach out to your physician. And for this one, I'd like to invite Hussam to join the conversation, specifically to address those who have received gene therapy previously as it relates to our program.
Yeah, thank you, Beth. Happy to answer that question. Yeah, in terms of prior gene therapy treatment, that's not an exclusion. At the same time, those patients do also have to meet other criteria. So please reach out to your treating physician who can assess that criteria and see if you're eligible.
Great. So if we're on a current therapy, definitely reach out, and there may be opportunities to pursue the program as well. Okay. Does this managed access program impact your current phase 3 trial in any way?
Yeah, I'm happy to answer that question. So as part of the accelerated approval pathway here in the US, we also plan on conducting a phase 3 trial. At this point, the MAP program that we've shared this morning doesn't really impact the phase 3 trial. That is moving forward. We're still kind of in the planning stages of that. So can't share a lot of specifics in terms of the geography, in terms of where that is going to be established and available. That is forthcoming.
Great. Thank you so much. And I promised last topic, and then I will hand it back over to our intended hosts and guests. In terms of the idea of coverage around a managed access program, is it correct to understand that the drug is provided by the company and that it will not have to be covered by individuals participating or their coverage providers as long as they're part of the program?
That is correct. Avidity will be covering the cost of del-zota, but we would like to mention there are routine labs and things of that nature that you would experience during a standard routine visit, and those will be taken care of as you usually experience, but Avidity will be covering the cost of del-zota.
Okay. And so in terms of infusion-related costs or anything, are those part of the managed access program, or are those costs that would be outside of the program?
It's a great question. I know we're providing del-zota, so I can get back with clarification. Any kind of outside costs outside of the program would not be included, but we're happy to support the community in providing access to del-zota.
Okay. Again, I appreciate you allowing me to interrupt. As a reminder, this program is for individuals who are amenable to exon 44 skipping living in the US. Again, we read the release this morning with such enthusiasm and a deep appreciation for your commitment, sort of your acknowledgement of the need that exists, and really, this novel way in Duchenne to help get a product into individuals who have been waiting. So thank you so much for your commitment. Thank you for this approach. And Rachel, Dr. Panda, I apologize, and I will hand the microphone back over to you. But Beth and Hussam, we really appreciate you allowing me to steal some time today.
Can I just add one more thing about the map, just in general? Thank you, Hussam and Beth. I was excited to see the news that came out today. I just want to, and thank you for clarifying that there is a process involved with the providers. It's not just that it's available already. But I also want to just highlight that all of these MAP programs do require IRB approval and things. So I think that's also involved. I just wanted our families to be aware of that. So if you reach out to your physicians, it is going to be a process in terms of starting this paperwork, getting IRB review approval, and all of that.
Thank you, Dr. Panda. Yeah, and additionally, if you have questions, please do reach out to your doctor. And as we have the website available that'll have inclusion criteria, we'll make sure that's shared. And then please do reach out to me personally if you have questions. You can also reach out to patients@aviditybio.com, and you can have your questions answered that way as well. So we're accessible and just really excited to bring this program to the Duchenne community. So thank you again, Kaylin, for taking time to acknowledge the press release.
Great. Thank you so much, Kaylin, again, for bringing up those great questions. We'll go ahead and pivot now to a question and answer session as it pertains to Beth and Dr. Panda and Hussam's earlier presentation. And I think a good place to start might be just seeking some clarity around regulatory pathways and different phases of clinical trials, because we've had a conversation about MAP, which I think is super useful and helpful. But as I look at the questions that are in the chat, I'm also seeing questions about, well, can I be on the clinical trial? Or when will I be able to access the phase 3? Or how is the phase 3 trial different from an expanded access program? So I wonder maybe, Beth, Hussam, if you could just sort of clarify what these different general pathways are and which ones are accessible in this moment and moving forward as we think about moving towards accelerated approval.
Yeah, thanks, Rachel. I can address that question. So the phase 3 program and the study is really a regulatory requirement for these molecules that pursue accelerated approval. So the accelerated approval pathway is well established in the DMD space for exon skipping therapy. And that is, utilizing the primary endpoint of dystrophin production. And you saw the substantial amount of dystrophin that, we've demonstrated in the EXPLORE44 trial that Dr. Panda just reviewed. As part of seeking accelerated approval, there is a requirement to also conduct a confirmatory trial. This would be a placebo-controlled trial with more patients looking at functional endpoints. And that would be something that would be ongoing at the time of accelerated approval in the US. In terms of the managed access program that we just reviewed earlier, this is really in the US to kind of bridge, potentially to allow patients to gain access outside of a clinical trial in anticipation of potential approval in the US.
Great. Thank you so much, Hussam. And I think a follow-up question to that might be related to all of the questions about access outside of the US, particularly in that phase 3 trial. Is there any expectation that there would be trial sites outside of the US for that phase?
Yeah, certainly. I think this conceptually would be a global trial that we're thinking through. And as part of that, there would be access outside of the US as part of that phase 3 trial. We're still working through a lot of the details around the specific countries and sites. And so we're still working through those details.
Great. Thank you so much. I think it's so helpful to understand what the forward vision looks like, even if those details aren't in a place where we're able to talk about them in specifics. So I'm really grateful for your response, Hussam. A related question, I see lots of comments in the chat about lowered age limits, whether it be through the clinical trial or even, post-regulatory approval and commercial access. Can you speak to any future plans to lower or change the current age banding requirements?
Yeah, in terms of the current trial that we have ongoing, which is the EXPLORE44-OLE, that study is fully enrolled, so it's not accepting any new participants. In terms of the MAP, as Beth mentioned earlier, There is a lower limit of 6 years of age, and that's primarily due to the availability of the safety data that we have at this point to support that. In terms of accelerated approval, we can't really speak directly to that at this point in terms of, the potential requirements there. At the same time, there is precedent in this space that there may be access to lower age limits, but this is a process that we have to work with the agency on.
That's great. Thank you so much, Husam. Another theme that I'm seeing within the chat is just additional question about genetic variant and different deletions or perhaps duplications that would be amenable to exon 44 skipping. So, Dr. Panda, I wonder if you could perhaps talk a little bit about just the concept of skipping, perhaps if we're able to pull that slide up and kind of talk in more detail about deletions that would be amenable versus those that would not.
Sure. Beth, if you want to go back to I think you have a tool that can also tell, but if you want to go back to the where Husam explained about the puzzle slide, I think here we are specifically talking about exon 44 skipping. Now, just kind of before we get there, I just like Husam explained here, when you look at this DMD gene, right, it's one of the largest gene in our body, and it has we call these each individual piece exon. It has to have these 79 pieces, and they're all different shapes, as you could see here, and they all kind of arranged in a way that they fit a puzzle together. So it has to look exactly like this for this gene to function properly to make this full-length dystrophin protein, right? So I'll get to the 44 in a minute. For example, let's say someone has, as we know, most common mutations that we see in this gene is deletion of these exons, meaning the piece of these one or two pieces of this are missing, and that we call them deletion, right? So let's say when someone has exon 45 deletion, can you point at 45 using your laser?
Yes, I can.
Okay, I don't know if you see that. Yeah, so 45, let's say if that piece is missing, right? Now your 44 and 46 cannot fit the puzzle together, like Husam was showing in another slide. So meaning your puzzle is not, fitting together. So the process, everything stops at 44 right here, and your body is not making any protein. I think that causes Duchenne, right? What is this skipping does is, like Husam was saying before, that medicine is going to hide this 44. So you already have the 45 that's missing, and it's going to hide the 44. Now the 43 and 46 can fit the puzzle together. So they're kind of restoring the reading frame. Now, I don't remember by heart, like what are the deletions, but you have to have deletions in that region that would make you amenable, like they'll make you, so you could skip that 44. Duplications sometimes would be also okay, but for certain skipping, most of the time it's deletions would be much more common ones. So Beth, correct me, is this all of these deletions that are amenable to 44? They listed here?
That we're aware of at this time, yes.
Okay, got it. So yeah, you can look at this all. Any duplications that we know of that would be amenable to 44?
Yeah, I mean, there is the 44, duplication.
Yeah.
And conceptually, that would also be amendable to this potential therapy. In our clinical trial, we obviously, have included, a number of different genotypes as depicted here. But obviously, it's a fairly, small sample relative to the entire scope here of all 46.
Right. I think that that's really helpful. I know that this is a challenging concept to really wrap our heads around, even with exon skipping in other areas of the gene being FDA approved. This is new still. And I think, as we consider a different exon that we are now considering skipping, it's helpful to sort of reframe and refresh our background knowledge on what's eligible and what's not and how these things all work together. Another question that I thought was really helpful in understanding the boys that have been treated to date is that of background steroid use. So I think this is probably a Hussam question. But Hussam, can you tell us about the steroid regimens of the participants to date? Have they all been on daily? Have we seen a split of daily and weekend or intermittent dosing? And has that perhaps led to any differences in data that we might expect to see?
Yeah, sure. I can answer that question. Before we get off of the slide, I do want to really highlight the importance of patients and their caregivers knowing their deletions, right? Because that really is the first step to identify what potential therapy may be available. I think that's probably the key message for me is really understanding what that deletion is to be able to identify potentially a path forward. In terms of the steroid use, the criteria within the EXPLORE 44 and OLE studies was really that they had to be on stable steroid use, I believe, for at least 3 months. But there wasn't any specifics in terms of the type of steroid or the regimen. It was just stable therapy on steroids. Dr. Panda, feel free to add to that as well if you'd like.
No, agree. Yes, there was no specific, and there were some who were not on steroids, but 90% of the participants were on steroids in the trial.
Great. I think that that's really helpful. I wonder, maybe Beth, you could take us to, let's say, slide, let's go to slide 24, if you could, about the four-stair climb. And this is more of sort of an in theory and just like data background question. I was really taken aback by all of the data presented today. It really is incredible. And I think one point of clarification, as we think about seeing a 2-second decrease in the time to complete in this slide, the four-stair climb, is really remarkable. But I wonder if you could help me understand, is this 2 seconds quicker compared to natural history, or is this on an individual patient level, they themselves are 2 seconds quicker than they were 1 year prior when that baseline assessment was completed?
That's a great question. Can I take that? I'm sorry that I didn't clarify that during the presentation, Rachel, and thanks for calling it out. It's actually compared to their baseline.
So this is each individual participant on average got 2 seconds faster than they themselves were the year prior.
Prior, yes.
Great. Okay. I think, we saw similar results with the time to rise and with the 10-meter walk run as well. And so I want to highlight that because I think sometimes as we look at these graphics and as we look at these data, it can be difficult to discern what we're comparing that data to. So thank you for just reinforcing that concept, Dr. Panda. I think it's really helpful to understand. Okay, I'm going to go over to my chat and see what other questions are coming in. I know that we've got a really robust chat going on right now. I see lots of questions about the MAP program, which is really exciting. I see lots of questions about access all over the world, which we've talked a bit about. I think another really practical question here about del-zota is, what's the frequency of dosing? Is it daily? Is it weekly? Is it monthly? What could an individual and their family expect if they were to gain access to del-zota?
Again, I think right now the dose that's being studied is 5 milligram per kilogram every 6 weeks. I think that's what I would expect in the managed access program as well as moving forward.
Great. And is there a loading dose with that where you have to receive treatment more frequently at the beginning before going to every 6 weeks, or is it every 6 weeks from the time of initiation?
No loading dose every 6 weeks from the beginning at the time of initiation.
Okay, great. Thank you so much.
Jump in if you need to add anything.
Yep, no, it's every 6 weeks, and that's essentially what has been evaluated. So there's certainly the efficacy data in terms of dystrophin and some of the preliminary functional data that has been observed, as well as the safety data that has been shared, is based on the 5 mg per kg every 6 week.
Can I ask a question, Huzam and Beth? Going back to the map, if you're able to share, I think where are you envisioning the infusions to happen? Are there going to be room for like home infusions, or is this going to be in an institution? Has to be a neurologist who has to prescribe? Can that be a primary care doctor who could prescribe that? Not prescribe meaning ask for MAP. If you're able to share, if you have details right now, if not, sorry.
Yeah, no, happy to speak a little bit to that, Dr. Panda. Really, I think the best options here for patients, and the first thing for them to do is really reach out to their primary care physician, and really kind of look into that. There are criteria, of course, in terms of the administration and the different types of institutions that would be required. I really can't go into specifics now, but I think that would be the first step. Now, obviously, this is an IV infusion, so having this at a facility that could accommodate that is going to be critical.
Thank you.
Thank you, Hussam. I think that that's a really helpful piece of information to take with us as we consider what this program looks like in sort of that lived experience. I have another question here that is really great, and it speaks to some of the different outcomes measures that are used in the context of the clinical trial. But the question is, have we seen any cognitive or behavioral improvements with the trial patients? So I wonder, maybe if we don't start with Dr. Panda for that, maybe you could just remind us what we are measuring in the trial participants, and kind of go from there as we think about cognitive impact of drug.
Sure, I can talk about. So we are not measuring any cognitive outcomes in the trial. Mainly, we are focusing on dystrophin expression and as well as the functional outcomes. Now, it may be a question for Hussam, or are we expecting this drug to go into CNS to have any effects? I don't know if you have any thoughts, Hussam, on that.
Yeah, so as I kind of showed in the schematic in terms of the technology, really the primary sites of action are skeletal muscle as well as the heart. And in terms of penetration into the CNS, we haven't really been able to demonstrate any meaningful activity there based on preclinical data.
Great. Thank you so much, Hussam. I think that this is an area where there's just such an appetite for more information. And really, we are hearing from the community that they're hopeful to see a future where additional types of outcome measures are included in clinical trials. And so that's something that PPMD is thinking a lot about right now, and engaging with key opinion leaders on what this could look like as we learn more about drugs that do cross the blood-brain barrier, and how we can really think about monitoring some of these outcomes that might be outside of the typical skeletal muscle measurements with which we're already so familiar. I do want to go back and just clarify one quick point. Hussam, when we were talking very briefly there about the MAP program, you had mentioned referring to the primary care provider doctor to start that conversation. When you say primary care, are you thinking the primary neuromuscular provider or like the pediatrician?
Correct. Yeah. I mean, I think it's probably the neuromuscular specialist, maybe the kind of direct physician. At the same time, I think participants should and patients should really kind of reach out to their doctor, whether it be a neuromuscular specialist, pediatrician. That's really the best way to kick this off.
Great. I think that that's really helpful. So what I'm hearing is go to your neuromuscular team. If this is something that you're interested in, that's a really great place to start. that there might be a role for your primary care pediatrician, but that's likely going to be in consultation with your neuromuscular team, working together as that multidisciplinary team of providers to provide care for the child or the individual interested in receiving treatment. So thanks again for clarifying that. I've got another question here about fluid-based biomarkers. So this is probably a Hussam question. Were there any other fluid-based biomarkers that were assessed or in which you saw changes in the OLE or the Explore 44 trial?
Yes, and Dr. Panda can speak to this. We've shared this data in the past in terms of other markers of muscle damage, including ALT, AST, myoglobin. So these were also consistently reduced in the same way in terms of the CK data, where they went down to near normal levels. Dr. Panda, feel free to elaborate on that as well.
No, I agree. As soon as I saw the CK data, I'm like, I don't need any other biomarkers, just a dystrophin and CK. We don't see this, right? CK going down to normal in this disease. Again, it may be the protein that's being made, being exon 44 skip amenable patients, but you don't see that. I think I'm impressed with just that CK, and that's something that we clinically look at, we use all the time. So I wasn't paying more attention to the other ones, but I agree. I think they were also in the I don't want to say I didn't pay more attention. I wasn't curious about those after looking at the CK.
That's great. And maybe Beth, you could actually bring that graphic up that shows the line over time with CK levels and how it's being monitored. I think that that's really your money shot, if you will. That's just it's really remarkable to see that because it's so unlike things that we've seen before. And so I want to highlight this again. And I think, worth clarifying too. The first time that I looked at this graphic, I was actually a bit confused. So I want to clarify that gray box that you see there, and please correct me if I'm wrong, Beth, Dr. Panda, Hussam, but that's when folks that were on placebo in Explore 44 went on active drug. So you saw them with that elevated CK, and then, yep, right where that marker is, we see that decline because that's when they're initiating drug. Is that correct?
Yes, you're nicely pointing out what I missed, Rachel. Thank you. Every single time. Yes.
I was like, wait a minute, they're on placebo in the gray blocks. I don't understand how folks on placebo had a decrease, but I
Yeah.
think that it's always worth just reminding folks of some of these elements so that we're all speaking the same language.
Yes.
Okay, let me see here. I think one other theme that I am seeing in the chat and through some of the questions that were submitted in advance were about other products in the pipeline and Avidity. And so I'm not sure how much we can speak to it here today, but I wonder, Hussam, Beth, if you could provide just a quick update on development of exon 45 skipping, just because I think that there's a lot of excitement.
Yeah, no, we're also very excited to move the AOC 1045 program forward. As I mentioned earlier, this is at the preclinical stage right now, so really preparing all of the preclinical work to enable regulatory filing. Beyond that, I can't really share at this point around timing. Um, uh, so that, that's as much as I can share today.
I think that that's great. And I think that, sometimes when the answer is vague or nondescript, that's helpful to understand that that's where we are, that we can't talk about these details just yet. There's just so much appetite and excitement there. I have two last questions for the group before we wrap today. The first is, what are some of those risks of treatment for families who are considering this therapy for their child or for their loved one? Maybe we could look at that slide that had the biggest or most commonly seen AEs. So I don't know, I don't know if this is a Dr. Panda slide or
Mm.
a Sam slide.
Yeah,
Yeah.
please, Dr. Panda. Yeah.
Sorry, go ahead, Hussam, if you want.
No. I was just gonna, I think if you could review this, this would be important.
Yeah, sure. I think from a safety standpoint, we call them TEAE, meaning treatment emergent adverse effects, but most of them are most common where we define that as occurring in more than three participants, and mainly those were procedural pain related to IV and headaches, and they were mild. The other ones that we saw in the open label were the upper respiratory tract infection, diarrhea, fall. I don't think any of them are related to the study drug itself. There was only one that was related to the study drug, which we talked about, was the allergic hypersensitivity reaction. That was so the patient, the participant discontinued from the study. So other than the allergic reaction, I don't think there was anything that was actually related to the drug itself, if that
Great.
helps.
I think that's incredibly helpful. We want to look at it from the whole picture, right? So I think the pros are very obvious as we look at some of this early top line data, but understanding sort of the flip side of what somebody might experience is also super helpful in sort of weighing risks and benefits. And then my last question, and I recognize we're a couple of minutes over. Maybe Beth or Hussam, if you could just remind us overall major time points over the next coming months as it relates to the MAP, the phase 3 trial, the accelerated approval engagements with the FDA. What are some of these key dates that we should keep in the back of our mind as we look toward a hopeful and eventual FDA approval of del-zota?
Yeah, I'm happy to address that question. So as Beth articulated, the MAP program is open. And it's going to take a few days here to get all of that information on the clinical trial.gov website as well as our own website. But it is open, and patients can reach out to their physician in that regard. We are, working through the phase 3 program, which is a requirement to achieve accelerated approval. And we'll be able to share more information around that moving forward. And we are still on track in terms of a BLA filing next year in 2026.
Great. It's an exciting year ahead for Avidity and for the Duchenne community. Beth and Hussam and Dr. Panda, thank you so much for joining us today and presenting this top line data, helping us to understand the map and answering all of our questions. I think that this is just incredibly exciting and is really, a piece of good news as we head into the Thanksgiving season here in the US. We're incredibly grateful for you taking the time to update us with all of this information. As a reminder to all, this was recorded and will be available on our website if you want to go back and rewatch it at a later date. And if you have any questions, please feel free to reach out to the PPMD team and we can direct you to the appropriate individual depending on the nature of your question. Thank you so much for joining us today and spending a part of your afternoon with us. And with that, we'll go ahead and conclude today's webinar. Thanks so much.
Thank you.
Yes.